Hello and welcome. Today we're going to learn a fundamental skill in biology, how to draw cells from microscope slides and photomicrographs. By the end of this lesson, you will be able to draw cells accurately and according to scientific guidelines. You might be wondering, why do we draw cells? Why not just take a picture? Drawing is a powerful tool for learning. It forces you to observe the cell's structure, organization, and the relative sizes of its organelles very carefully. This active process of observation and representation helps you understand the cell's anatomy much better than just looking at it. In other words, drawing isn't just about creating a picture, it's about training your eye and your mind to see and understand the microscopic world. Now let's go over the essential guidelines for creating a proper biological drawing. These rules ensure your drawings are clear, accurate, and scientifically sound. First, always use a sharp pencil. This is crucial for clear, fine lines. You should never use shading or coloring in a biological drawing. Second, your lines must be clear and unbroken. Avoid sketching or using fuzzy lines. Each line should be single and clean. This makes your drawing easy to interpret. Third, maintain correct proportions. This means drawing the cell and its organelles to scale, reflecting their realistic size and shape. For example, don't draw a nucleus that's half the size of the cell if it's actually much smaller. Fourth, and this is very important, no shading. Instead of shading to show texture or depth, you should use label lines to indicate structures. Shading can obscure details and make your drawing unclear. Fifth, labels. Label lines must be straight, horizontal, and drawn with a ruler. They should all be on the right side of the drawing and form a vertical list. The words should be printed and easy to read. This creates a neat and organized drawing. Finally, every drawing needs a title. The title should be informative, mentioning the cell type, its scientific name if known, and the magnification at which it was viewed. A good title tells the viewer exactly what they are looking at. Let's look at a great example of a biological drawing, a human cheek cell. First, notice the title. It's informative, including the scientific name, Homo sapiens, and the magnification, 400x. It's important to state the magnification of the microscope, not the magnification of your drawing. Next, observe the cells themselves. They are drawn with clear, unbroken lines. Instead of shading, a technique called stippling is used to show the texture of the cytoplasm. This is a good alternative to shading if you want to add some detail. Now look at the labels. They are all on the right side, forming a vertical list. The label lines are straight and drawn with a ruler, and they do not cross each other. This is the correct way to label your drawing. Finally, notice the scale bar. A scale bar is essential because it shows the actual size of the object you've drawn. This allows anyone looking at your drawing to understand the true dimensions of the cell, regardless of how large you drew it on the page. The drawing also includes a calculation for the drawing magnification. This is the ratio of the size of your drawing to the actual size of the object. It's a good practice to include this calculation to show the scale of your work. Let's compare the common structures you'd label in a plant cell versus an animal cell. For a plant cell, like an onion epidermal cell, you would typically label the cell wall, cell membrane, cytoplasm, nucleus, and often a large central vacuole, if it's visible. Here's a diagram of onion epidermal cells. Notice their distinct, rectangular shape, which is due to the rigid cell wall. You can clearly see the nucleus and the large vacuole pushing the cytoplasm to the edges. Now, for an animal cell, like a cheek cell, the list is shorter. You would label the cell membrane, cytoplasm, and nucleus. Animal cells lack a cell wall and a large central vacuole. This difference in structure is a key distinction between plant and animal cells. Plant cells have a rigid cell wall for support, while animal cells are more flexible and closed only by a cell membrane. Now let's talk about the process of observing and drawing from a microscope. First, always start by focusing your microscope using the low-power objective. This gives you a wide field of view, making it easier to find your specimen. Once you've found it, you can switch to a higher power objective for more detail. Second, observe the structure carefully. Pay close attention to the overall outline of the cell and the position of its organelles. Don't rush this step. Good observation is the key to a good drawing. Third, sketch the shape. Start by drawing the outline of the whole cell. Only draw individual organelles if they are clearly visible. Remember, your drawing should reflect what you actually see, not what you think should be there. Finally, add your labels using ruler-drawn straight lines, following all the guidelines we discussed earlier. This completes your drawing. Sometimes, you'll be asked to draw from a photomicrograph, which is simply a printed image taken through a microscope. Here are some tips for drawing from photomicrographs. First, remember that you are drawing what you observe in the image, not copying it. Your drawing should be a simplified, clear representation. Second, don't include unnecessary background details. Focus only on the cells and the structures you are asked to draw. A clean drawing is a good drawing. Third, and this is very important, keep your drawing large enough to show detail. A good rule of thumb is that your drawing should take up at least 50% of the available space on the page. A small, cramped drawing is difficult to label and interpret. By following these tips, you can create accurate and effective drawings from any photomicrograph. Let's quickly recap what we've learned today. We started by understanding that drawing is a powerful tool for learning and observation in biology. We then covered the key guidelines for biological drawings. Use a sharp pencil, clear lines, correct proportions, no shading, proper labels, and an informative title. We saw how to apply these rules to draw a human cheek cell, including the use of a scale bar and the calculation of drawing magnification. We compared the structures found in plant and animal cells, noting the presence of a cell wall and large vacuole in plant cells. And finally, we discussed the process of observing and drawing from both microscope slides and photomicrographs. You are now equipped with the knowledge to create accurate and effective biological drawings. Practice is key, so get out there and start drawing. Thank you for watching.